Okay, I've got 3 o'clock. There are 45 of us here so far. We'll have a few more joining us, I think, but we will get started. We've been planning this for a while. I think Pamela sent the email in November that said, should we partner and have a cross-site data nugget online program? And Liz and I said, yes, absolutely we should. So that is what brought us here today. So kudos to Pamela. I'm very excited that she started that idea. So just some group norms and reminder, we're still in a not normal world. So don't apologize for where you are, where you're learning from, where you're working from. I'll ask everybody to make room for many different voices. Embrace curiosity, but question ideas and not people. Use the chat as a placeholder for thoughts or questions. I'll moderate that the whole time we're together today. And no one knows everything, but together we know a lot. So it's wonderful that there are so many of us gathered to think more about climate change and plants today across the country. So Zoom norms, we're recording so that we can share it with you and others. I will say that the breakout rooms will also be recorded, but we won't publicly share the breakout room conversations, but we will more publicly share this first part of the program today. Remember, you have some options for how you want to view the program today, speaker view or gallery view. And everybody's doing a great job of being muted when not talking. Your video is encouraged today for participation, but if your internet's slow or we sound glitchy, feel free to turn it off. Or if that supports your learning better to have your video off, we acknowledge that that is fine too. And just a reminder that the chat, even the private chat between 2 people, it all gets saved. So just keep that in mind. Here's our agenda for today. We've packed a lot into this agenda while also giving work time for us to work through and read the data nuggets and be reflective on them. So this is the first time we've done this program, so perhaps we've bitten off more than we can chew, but we have put a lot of thought into it, and we're going to see how it goes. We think 2 hours is a lot to ask of an online program, but hope that it gets the right mix of participation and listening. All right, so let's meet everybody. I've been chatting for a moment, but we want to hear from you, so feel free to use the chat. So my name is Haas, and I work for the Kellogg Biological Station up in Southwest Michigan. We use the hand, right, in Michigan to show where our cities are. And I oversee community outreach across the biological station and work with our LTER to provide professional development for K-12 teachers and student experiences related to our science. I'm also a doctoral student in science education, interested in informal, formal teacher partnerships, much like the type of program we're having today. So that's me, and I'll pass it over to Pamela to introduce herself.
Hello again. I'm Pamela, and I'm very excited to share with you as much as we can about data nuggets from Harvard Forest LTER, which is located in Petersham, Massachusetts. I work with teachers primarily from grades 4 through 12 in leading field ecology projects, as well as other ecological content related to the studies that our professional ecologists here at Harvard Forest are doing. You're going to hear from one of those in a little bit. Welcome.
Right, hi, I'm Liz Schulteis, and I have my PhD in plant biology, but I got sucked into science education just because I enjoyed working with teachers so much, and it was such a, like a fun thing that I was doing as a grad student. And so I'm one of the creators of Data Nuggets, and through that work, I've been able to continue working with the LTR and continue working with teachers. So I'm excited to share some of the cool data sets that we have in Data Nuggets with you today. Okay, I'm taking over here. So I'm going to quickly just introduce, we also have with us Meredith and Neil, as basically our two, scientists experts from the two LTER sites. So Meredith is going to be giving our plenary talk and the background information on plants responding to climate change and setting up the theme for the workshop. And then Neil is going to introduce the research from Harvard Forest and the data nugget that they have from there. And then we're going to wrap up after the first hour. We're going to send everyone into breakout rooms. And here's all the names of the experts we have with you in those rooms. We have teachers and scientists behind the research and grad students, basically people doing any research related to the work in the data nuggets. So this is your chance to really ask in-depth science questions and get to know everything you'd want to know to effectively use these in your classroom. Okay, and we also, you all introduced yourself in the chat and said where you're from, but we have, your addresses to send you your care packages. So we put those on a map just so you could see where all the participants are coming from. We have a, wide range of everyone calling in. Here's the Harvard Forest site. Here's the Kellogg Biological Station site. And so we're just so happy that we have such a cool spread of people and everyone, is calling in from different places. Okay, so we're going to start off with a quick data activity with all of you. So you all should have gotten care packages in the mail. You got this tiny box of conversation hearts. We figured the event is near Valentine's Day, so let's send you all some hearts and some sugar to stay awake. So if you have those hearts with you now, take them out. And basically, open up the box, spill it out on the table, and we're going to do just a quick data activity with those. And if you ate them, that's also fine too. I don't have any with me, so you can still participate if you've already eaten yours. So, all right, yeah, spill them out on the table, start to pick around in the hearts and take a look at them. So we want to model an activity, a data activity you could do with your students in the classroom. So start to look at the candy hearts and think about what data could you collect from these. So treat it like a little data set. What are some variables that you could start to collect data on? So if you want to start typing in the chat, any ideas that pop into your head, feel free to put it in the chat. I'm sharing my screen so I can't see you, but you'll all be able to see each other's ideas in there. So
color, words, phrases, amount of each color, broken versus not.
Oh, awesome.
Sayings.
That's great. Yeah. So there's a ton of data you can collect on a pretty simple item.
Number of letters.
Ooh.
Good thought, Kelly. We didn't think of that one.
I love that.
How many words? Taste. The mass of each one. Oh, you guys are way more creative than we are. These are great.
Yeah, these are way better. Okay, so Kara, if you don't mind in the chat now putting the link to a spreadsheet. We have a collaborative spreadsheet set up for all of you. So I see a good amount of people are in there already, actually, so that was really nice. Basically, each of you have your name in this spreadsheet. So you can go find the rows where you can put your own data in. So search, if you hit Command F or Control F, depending on what kind of computer you have, you can find and search for your name. And you all have 10 rows to start typing in data. I want to put in these other ones that people had. So number of letters was a good one, and then broken or whole. So basically, just find your rows and start filling out your cells of data. So I can scroll through and just start to, I see people clicking around, so it looks like you're all finding your names. Some people are already typing, perfect. And then Kara, for color, created us a drop-down menu. So there should be just a finite set of colors that you have to choose from. But with the phrases, we've realized it kind of depends what box you got. You might have a phrase that nobody else has, which is kind of cool. All right, so I'm going to keep scrolling around.
Hi, my apologies. I'm just joining you. So how do I share this or get this screen?
Kara in the chat has put a link to a Google Doc. So just
Yep.
open.
And I'll reshare it, Jane, in case
Okay.
you
Thank you.
popped in earlier.
Thank
So
you.
it should be there now. Yep, no problem.
Great. Okay, so while you're all entering data, I can start to pull up a graph of the data set. And so you can see like this is a great opportunity with your students to start to brainstorm, what kind of variables can we collect? What kind of questions can we ask now that we've collected data on these variables? So you can start in the chat too, you could start to brainstorm some questions that we could ask with the data. And then based on the questions you come up with, that will affect the type of graph that you want to make to show the data and answer the question. So this program I have open is called Data Classroom, and we can send out information about the program to all of you later. It's nice because it links to a Google spreadsheet and you just have to hit refresh and it will bring new data over. So I'm going to refresh it now to see how it's changed now that you've all started putting your data in. I'm going to close it and try again. Here, I can show you, this is the graph that we had before all of you started putting your data in. This was just based on Kara and mine, but we have, we can start to explore the question about the number of different variables. So how many pink are in the box, how many orange are in the box, and we can make a histogram which shows the frequency of these different colors. So you could use this to explore the question, which color is the most common across boxes? So it would look like orange was the most common between Kara and mine. Let me try to refresh again and see what happens. Maybe the error is because we're all entering it simultaneously. So let's see what happens.
It's not letting me enter because it says there's too many people in there.
Oh, we broke it. All right. Well, we couldn't test that because we didn't know, or no error came up when so many people weren't in.
Let's just take a pause. Liz, you want to just try a pause on data entry and then hit recreate the graph and see if it'll. So let's take a pause on data entry on the Google slideshow and just give it one more try, Liz, to see if Data Classroom will load it.
Awesome. I was thinking too, maybe I'll try to make a copy and see what happens because none of you would be in the copy, but let me hit refresh one more time. I will say maybe if your classroom is too big, I would imagine it should be able to handle a classroom of about 30 students. So we overtaxed it with 50 people.
Yeah, there's more. There's 63 of us now. Okay. So I wonder, I don't know.
Anyway, you see what the graph would have looked like. And what's really nice is you can kind of toggle through all the different variables. So we can do phrases. So here's all the different phrases and the frequencies of how many were in each box. But yeah, so the data would become a lot more interesting, I think, if we had a lot more people in there. But okay, well, maybe what I'll do is at the end of the event, I will reload this in the background and then I'll share our graph. So if you want to keep entering your data into that spreadsheet, I'll refresh it. As you get more data into the program, you can do different kinds of graphs. And then the graphs would basically allow students to say, what kind of question do I have? This graph lets me answer that question, and you can, play around with the data. So once you've entered all your data, feel free to eat your candy too.
That's right. Enjoy your candy. Thank you for playing along.
Yeah, thank you.
Breaking the ice with us with Conversation Hearts. And then on to Pamela.
So Liz, you're okay. Thank you. I'll start sharing now. So here we are. We're going to do a little overview of what is LTER. Kara and I work for the National Science Foundation's long-term ecological research network, where we are two of 28 sites in a national network doing long-term ecological research. Kara and I are outreach people, and we have Meredith and Neil as scientists here, and we have a few of the other education staff with us today. So you saw on the map where you're all from today, and now you can see another map to see where the 28 sites are located for the LTER network. So here we are, Harvard Forest to the east in Massachusetts, and KBS right there by the Great Lakes in Michigan. We have a number of biomes and ecosystems represented that we're studying at these different sites. You can see that I've featured some in the Antarctic down below here, and you have some in the Arctic, you have some in alpine sites, deserts, prairie. Kellogg Biological, who's with us today, is an agricultural site. We're a forest, and among other forest sites as well. We have some ocean sites, both at the coast and offshore. So real wide range, all collecting long-term ecological data. Here's some images of just a few of the sites. It's really hard to pick and choose because there's so many interesting sites, but to give you a range of different kinds of sites and what they look like, aside from our own sites, which we're going to highlight in a minute. So long-term observations that we're maintaining long-term records of the ecosystems. There's a lot of long-term experiments, experimental manipulations showing change over time, long-term relationships that we're building with resource managers, educators, and landowners in our region. And we're bringing diverse groups together to do this work. And of course, we're all working to get a better understanding of the inherent variability of natural systems. Harvard Forest, as I mentioned, is located here in Massachusetts. We're in central Massachusetts, where this red dot is. We collaborate, however, with scientists from throughout the region. Some of the focus of our work is how ecosystems work. Trees, plants, soil, atmosphere, water, and wildlife are all things that we're looking at. And we're really looking at how ecosystems change over time and how our landscape change over time and how it all works together. This is a long-term plot, mega plot with thousands of stems. And this is what it looks like on the ground, the measurements. And these are some of the graphics we come out with in order to interpret those thousands of stems and what the story they're telling is. We're going to hear more from Neil Peterson a little bit later about other ways we're looking at change over time, and particularly how the climate change story, which you'll hear more from Meredith as well. Kellogg Biological Station is affiliated with Michigan State University in Hickory Corners, Michigan, right here where their star is. And they're looking at how can we farm the land in a way that helps farmers produce enough food, fuel, and fiber, and benefits the environment and the rest of society. So it's a unique site looking at agriculture as an ecosystem. We at Harvard Forest also have a farm, which is a fairly new part of what we're doing. So one way we can connect. Today, we're focusing on data nuggets. And so most of you, I think, know what data nuggets are, but some of you don't. Liz, who did that wonderful conversation hearts activity for you, is one of the 2 data nugget staff who leads us in this work of helping helping first of all develop and then share free activities that bring real data into the classroom. And a big part of it is the messiness and complexity of the data, that it's based on contemporary research written by the scientists. It's not always written by the scientists, but with the scientists involved in that process. Reveal to students the nature of science and research. And we all follow a familiar template when we're creating these data nuggets, which allows hopefully you teachers to apply it with some consistency and gain familiarity and have an ease of applying it, we hope. But we're also given a little bit of flexibility with how to implement those. This is a sample of what a data nugget might look like. If you look on the website, they're very pretty. And here's a sample of a piece of the table that would tell you what the title was, some keywords, a summary of the science, the content level, and the study location. So it's really well organized on the website, and that's why I guess Liz has thousands of you teachers on her email list, and a lot of people wanted to attend today's workshop because I think this is proven as a popular activity for teachers. And this is how students are working with this. They're spending more time doing science, and they have an improved ability to construct explanations, increased confidence with data and science, and increased motivation to pursue STEM careers. I'm going to be turning it over now to Meredith Zettelmoyer, who is from Kellogg Biological Station, LTER. So Meredith, are you ready to share your slides?
Yes, just give me one moment.
Okay.
Okay, so hi everyone. Today I'll be talking to you about plants and how they respond to climate change. But first, to give you a little bit of background, so I recently graduated from MSU and worked at the KBS LTER for the last 5 years, studying the role of different environmental changes, including rising temperatures. I need to move everyone's faces. Hold up. There we go. On prairie plants in the area. So here's an example of, this is called hairy beard tongue, and this one is called a tall coneflower. And now I am a postdoctoral researcher at the University of Georgia, but I'm studying climate change effects on species in the Rocky Mountains. So I'll actually be working at the Niwot Ridge LTER starting this June, and we're going to be looking at this really cute cushion plant. So I've worked at a few LTERs at this point, and just kind of the research theme across those is how climate change is affecting plants. Okay, so IPCC, the International Panel on Climate Change, predicts that temperatures are going to be rising somewhere between 2 and 6 degrees Celsius by the end of the 21st century. And so plants are affected by this in many ways because they respond to the elevated carbon dioxide levels, rising temperatures, and changes in precipitation. So plants are really responsive to climate change. So today I'm just going to tell you about a couple of the ways that plants can respond to climate change. So first, our warmer temperatures and the increasing carbon dioxide levels can increase plant growth. And as plants grow more and more, they're going to basically act like straws. So the larger plants are going to need more and more water. And in some cases, particularly in kind of dry areas, this can actually cause river drawdown. So this was a river that the bed was actually up to this ridgeline here. And now the water level has decreased pretty drastically. And then plant growth also means that there's going to be more vegetation making the Earth's surface even darker. And so that can actually cause the Earth to absorb even more heat. So there's kind of a compounding effect where if we have this green or this brown surface, then we're going to have what's called a low albedo, and then more heat is going to be absorbed. Whereas if we have situations where there's whiter landscape with snow, ice, or sand, then we can reflect some of that heat back into the atmosphere. So in contrast to increased plant growth, in some situations, temperature stress can lead to decreased growth instead. And this is a really important response, particularly for agriculture, because it can result in lower crop yields. So here you're seeing a cornfield where the plants are really stressed out by temperature and are probably not going to produce as much in this year. And then similarly, as a response to drought, less water will also decrease plant growth. So you'll end up with these landscapes that are brown and dry and nothing's really growing. And this map here is showing the effects of the drought in California. So in 2001, this basin area had a lot of green vegetation if you looked at it from satellite images. And then in 2016, they imaged the same area. And you can see both that the vegetation has kind of recessed back from this water source, and it's turned really, really brown in this area. So plants, even though I just showed you they can increase growth in response to climate change, they can also cause decreases. So pretty variable effects. And one of the ways that scientists can kind of figure out how climate change is affecting plant growth, either in a positive or a negative way, is by looking at tree rings, which is something that Neil's going to introduce more in his introduction. So studying tree rings is called dendrochronology. And we can use this because tree growth is really sensitive to local environmental conditions. So looking at the rings in their trunks can give scientists information about the area's local climate over time. And so these rings can tell scientists the tree's age just by the sheer number of rings. And then the width of the rings can tell us about climate change. So the rings will be wider in warm or wet years. So here, this really wide ring that has the rainy season right by it, that means that this year was wetter. And then the rings should be thinner in colder and drier years. So like 2 years later, it looks like there was a dry season. And so because trees are so long-lived, this can tell us things about climate over thousands of years, potentially. And another way that plants respond really strongly to climate change is by shifting their phenology. So this is the timing of different life cycle events. So for a plant, this is going to be when you germinate, and then you grow, and then when you flower, and when they set fruit. And same as for these trees, phenology, so the timing of things like flowering, is really sensitive to local environmental conditions like temperature or precipitation. And then the timing of these events is really important for plant populations because it's going to get them access to resources as well as pollinators. And I'll give you more details about this when I introduce the springing forward data nugget. And most studies have found that many species are advancing their phenology to be early and earlier in the spring. So this figure is just showing a bunch of plant species from the US and the UK. And the species that are shown in red are species that are shifting their flowering and leafing earlier and earlier. So most species are becoming earlier. And you guys have probably noticed this if you see things like invasive species like multiflora rose often green up really, really early in the spring now. And the way that we often study phenology, there are a couple different ways. So we can look at historical records like we can for trees, but we can also do this experimentally by simulating rising temperatures. So for example, at KBS, we have this setup with heated rings where half of them are experiencing warmed temperatures by 3 degrees Celsius, and the other half we just let alone. And so students and researchers can collect flowering data in these heating experiments. And what we see, so I'm showing you an example with another beard tongue species. We can see that flowering day under normal versus heated temperatures, flowering day is often earlier when the temperatures are a lot warmer. Altogether, climate change effects on plants are really important because they can affect crop yield and food supply, particularly if the plants are temperature stressed or if they can't get access to their pollinators. If the plants are dying off, then that's going to be a lack of food for other organisms because plants are the base of that food chain. It can potentially cause decreased water supplies, can cause increased pollen allergies. So especially as plants start flowering earlier, things like ragweed actually can end up sending out more pollen. And then we also can lose species, particularly threatened or culturally important species, because of climate change as well. I'm going to pause here and we can take any questions. I can't see the chat, but maybe Kara can share any before I introduce the KBS data nugget.
We got a good amount of questions so far, a lot of them are about tree rings. And so Neil has been fielding some of them in there. So we got a question about if it's typical for rings to be thicker when trees are younger, and then why tree rings change color throughout the season. So Neil said that rings are wide when trees are young, and that cell size and wall thickness has to do with why the rings change color throughout the season. Okay, let me see. Neil, do you want to unmute and like share what you've been talking about with this with cell walls in there?
Yeah, it's mostly related, or it mostly applies to conifers like pines and larch and spruce. And in the early part of the season, foresters a long time ago called it springwood. And so the cells in the ring were larger to pass more water up to the tree so growth could get going. And the cell walls are generally thinner. And then later in the growing season, we call it late wood. It used to be called summerwood. The cell walls will get smaller. The cells would get smaller and the cell walls would get thicker, and that's a lot of the coloration that you see. That concept generally applies to oak and hickory, though not quite the same way, but generally that works. But it doesn't apply to maples and birch and other types of tree species like that. And I want to come back to the ring width variation because it's funny. It's sort of complex thing, and it can be very misleading to a lot of people, even PhD scientists. So when trees are young, they're generally small, though that's not always true. But when they're smaller, they're growing at a certain rate, and the rings are decently sized. You could say wide. And as the tree gets larger, the rings have to get narrower if it's growing at the same growth rate, mostly because of geometric constraints. So just putting the same amount of growth around an expanding stem will make the rings naturally narrower. But when we correct for that, that doesn't mean that the trees are growing slower as they get bigger or older. And in fact, we're finding that more to be the opposite. And so it's a complex thing that people notice. And yeah, we get asked that question a lot. And it's complex, but it's fun.
Awesome. Yeah, there's a lot of like, as we break out into our rooms to look at the two different data nuggets, there's a lot of math opportunities in both of these. So if teachers really want to, get into statistics and different math activities, feel free to ask the scientists some follow-up questions. Meredith, I have a question for you. Are there like standard, so one of the questions we got is if there's a standard like phenology date for certain species to compare against to know that like a year is particularly, early or late. So how would you kind of compare the phenology you're seeing in a year to like a standard?
So the question is whether there's a standard date for phenology. Not really. So what we can do is we can actually go and use herbarium specimens and get ones that are from, potentially before climate warming really started in a particular area, and try to get like an average day of flowering from that. But otherwise, the comparison is generally going to be like in this experimental setup. If you don't have some historical data set that you can get a flowering time from, you're going to have to use like ambient temperatures and normal temperatures versus some warmed, and that ambient will end up being your kind of control or reference date. So there's not a historical standard, no.
That's cool. Yeah, well, using herbarium specimens gets to the other aspect of their question about like region. So you could look for specimens specific to your region, that'd be cool.
Yes, you definitely can.
And then Mark responded that farmers and people who grow like apples would have historical data sets as well. Like, you could sort of rely on the knowledge in your area from farmers or other specialists. Let's see. Okay, people have a lot of tree ring questions. That's cool. People are getting jazzed about tree rings, so maybe Neil will let you field those when you're talking about the data nugget and introducing more about rings.
Sure.
All right, I think we're good on questions right now. People can keep using the chat, and Meredith is going to introduce the KBS data nugget now too. So she'll still, we'll have her as our specialist like throughout here. So keep, questions coming for her.
All right, so moving into the KBS data nugget, which is based on phenology. And so this data nugget is based on data that Jen Lau, Liz, Mark Hammond, and Sean Davis, who's also here today, collected. And so just super quick reminder that phenology is one of those critical ways that plants are responding to climate change. So I'm going to be talking today about just timing of flowering, but this applies to a whole bunch of other transitions in a plant's life cycle. And it's dependent on those environmental conditions. So why is phenology important? So the timing of when you flower really affects your growth and your reproduction, because it's going to expose the plants to different types of environmental conditions. So plants that flower way too early are going to potentially catch damage from frost. So for example, in Michigan, this happened with a blueberry crop one year where temperatures were warm, the plants flowered really, really early, and this is what happened, and the crop yields were a lot lower that year. Alternatively, if plants flower way too late, then they can catch that late summer drought period where they're not getting enough water, and then the fruits that they produce are not going to be viable to produce offspring and keep the population going into the next year. And then, as I mentioned earlier, phenology is also really important for helping plants match up with their pollinators. This is really important, especially for species that require pollination by a particular, so if it's a co-evolved situation where so for example, this iron weed relies on this species of Melissoides bee or early season bee for pollination. And if they are not matched up, this plant is not going to get pollinated at all. Phenology also affects what species get early access to a lot of really important resources like light, soil nutrients, and space. And then it can also affect reproduction down the line. So in this invasive species, researchers found that plants that flower really early, so on the far left of the x-axis, plants were producing a lot of seeds, but then as they flowered later and later, they were producing fewer and fewer seeds. So it can really affect reproduction later on as well. And altogether, because of this access to resources and declines in reproduction, phenology can really affect competition with other species. If you're a plant that gets in really early, so today we're going to be talking about invasive species, for example. So things like multiflora rose or garlic mustard that appear really early in the growing season, then they're going to get this huge advantage. And so as I showed you before, most species are shifting their flowering earlier in the season. So lots of these plants are shifting in red. But there are a handful that are actually delaying or not changing. So these guys in blues aren't responding to climate change in the way that we would expect. And something that this has interested biologists for a long time, and something that we've noticed is that non-native or invasive species tend to shift their flowering way more than natives. So this figure is showing you differences in flowering time between 1900 and 2010 from flowering that was recorded in Thoreau's woods in Massachusetts. And these researchers found that while natives were only shifting their flowering by about a day over time, non-native species in the area actually shifted by closer to 11 days per degree Celsius. So these non-natives were shifting their phenology while the natives were kind of just hanging out and doing nothing. And that early advantage can give them early access to all of those resources like space and pollinators, and give them a competitive advantage over those native species. So this data nugget looks at an invasive species in the simulated warming array at KBS on the LTER grounds. And so in this experiment, we have these plots in an old field grassland community. And these plots have heaters hung over them. And so we have half the plots are exposed to, here I'll show ambient temperatures first. So these have heaters hung over them, but they're not on. So they're just experiencing normal temperatures that they'd experience if we hadn't manipulated anything. And then in this other plot, these infrared heaters are turned on. And so they're warming the plot by 3 degrees Celsius. Basically by the temperature sensors in here are feeding information to this mechanical heating station that then tells these plots to be 3 degrees above whatever the blue plots are set. And so this experiment is set up with these 4 normal plots where the heaters are there but off. And then we have 4 heated plots with infrared heaters that you can see from this photo taken at night. You can actually see the warming happening. So what we had done is we planted a whole bunch of species where the species were either native, so like this brass Bromuscalmi is a native plant, and then in the same plant family we had it paired with a non-native species, so in this case the Dactylus. So we were able to compare whether flowering differed under these warmed versus ambient treatments in native versus non-native species. And so the way that we monitor phenology in these rings is just going out and recording data. So looking at what day it flowered, how many flowers there are, things like growth. We can get a whole bunch of data when we're out here. And so today, the metric we're going to be talking about is day of first flower, which is a really commonly used measure of phenology. And this is not when buds appear, but when you actually see the first open flower on a particular plant. And so in this experiment, I'm showing you the overall results first, and then we're going to narrow down to just one species. So this is showing you flowering that day to first flower in the native species and the non-native species under that ambient or normal plots in blue, and then the elevated or warmed plots in orange. And what we can see is that native plants flower in about 60 days after the start of the growing season, so after like the 1st of May. But they don't flower any earlier under elevated temperatures than they do under these ambient temperatures. Whereas the non-native species, first of all, they flower earlier on average. They take less than 40 days to flower. But under the elevated temperatures, they're actually flowering even quicker than they do under the ambient temperatures. So the native species are not responding to warming here, whereas non-natives are shifting a lot earlier in the growing season. One of the species that was planted into the rings as part of this experiment was Dame's rocket, or Hesperus matrinalis, which is a really common invasive species across North America. It is a member of the Brassicaceae, or the mustard family. So it's related to really common foods like broccoli, as well as a lot of invasive species like garlic mustard. And this plant was introduced to North America from Western Europe as a garden plant in the 19th century. And that's largely because, as you can see here, bees as well as butterflies and moths really like this species. It's a great source of nectar, and so they'll pollinate it pretty heavily. Dame's rocket is also invasive because it can survive in really disturbed areas. So open areas that have been, maybe we've come in and we've disturbed the soil, or we've cleared out some of the canopy. It also really likes hot summers, so climate change isn't likely to be a particular problem for it. And then it produces a lot of seeds, and also forms these really dense mats, so a lot of seeds from a lot of individuals. And so it started out in Central Western Europe in this map in green, but at this point it's expanded both eastward as well as westward, and now is a pretty ubiquitous invasive species across North America, the US and Canada at least. So this data nugget focuses on how temperature influences the phenology of Dame's rocket. And so what we'll be looking at is differences in the number of days to first flower. So again, that first appearance of an open flower between these normal and these heated plots. And this experiment is basically going to tell us under climate change what's going to happen to this invasive species. So these rings, the climate warming manipulation mimics predictions for climate warming in Michigan. So KBS is like here. So this is in the 2 to 6 degrees Celsius prediction range. So if Dame's rocket is flowering really early under our heating treatment, then that suggests that Dame's rocket might continue to flower earlier and earlier as temperatures get warmer, which should give it this competitive advantage over natives. And we might see similar patterns across a wider variety of invasive species.
Hi, Meredith. I'm sorry to interrupt you, but we're running low on time. Oh, but you're right at the last one. So perfect. Okay.
Good timing.
Yep. So
we aren't going to be able to field questions, however, right now. But if people want to write questions in the chat, as they have been throughout, if we find we have time at the end to address those questions, we will, or in the breakout group.
Pamela, I've actually been answering them, and sorry if I'm stepping on anybody's toes, as people are asking them based on what the author has written, Meredith has written.
Awesome. You're on top of it. That's good. Yeah, so Meredith, you can check out, now that you're not sharing your screen, you can see the chat again too, so you can see if there's any in there.
Great. So if you can stop sharing your slides, Meredith, I will go ahead and resume the slideshow here, and Neil can share his portion. Thank you very much, Meredith. That was really interesting because we're doing a lot of phenology studies at Harvard Forest as well. So I know a lot of my buds, leaves, and global warming teachers are here, and I'm sure they were very interested in that. Can you see that?
Yep.
Okay, great.
All right. I want to introduce myself a little bit more. I'm associated with the Harvard Forest and the LTER at the Harvard Forest, but I feel compelled to tell you that I am the product of 2 elementary school teachers. And when I was leaving high school, I was thinking that I was going to be a math teacher in high school, maybe be a lacrosse coach, something like that. And I never imagined doing what I'm doing now. And so this picture, I wanted to start here. This is when I'm in front, I have a mullet. This is from 1995 when I was a technician. Nope, stay on that. Nope, go back.
Sorry, I didn't realize I was failing here.
I was a technician working in the longleaf pine ecosystem of Southwest Georgia. And this is when I was starting to learn about climate change and learning about what its potential impacts might be and how our climate might change. But it was around this time when I was starting to learn and think about how it might impact forests and ecosystems, which is what I do today. And when I think about that time period, and I think about how little I knew about much. Next slide, please. So this is me just a few years later, and this is where I started really learning about climate change. I still had my mullet. I had my first beard, but this is in central Mongolia in the Hungai Mountains. And if you look over the tree, the top of the tree that's behind me, in the far distance, you can see Otontengger, which is a mountain glacier in central Mongolia, and a sacred area and an important source for drinking water. And we wanted to sample on that mountain to study climate change with the late Gordon Jacoby, who was my boss at the time. But we weren't allowed to go there because it is such a sacred area. We did, however, talk to a lot of the local people when we were driving around the mountains, and we were learning that it was they said, it's not raining, and it's very hot. And this is the first time that I had been in, this is the second time I had been in Mongolia. And I didn't know what they meant then, because to me, Mongolia is dry. And so I didn't see much difference between the prior year and that year. And what I know now, what we've learned through tree rings, is that was the beginning of one of the worst droughts of the last 1,000 years, just about. Next slide. But what we were doing, and this gets to some of the questions raised in the chat already, and thank you, Meredith, for giving the basics of dendro, because I wasn't planning on that, so I appreciate the background. We went to the mountaintop in central Mongolia, and this is Sologotin Dawa, or the Rainbow Mountain Pass is the name of the mountain, essentially. And what you're looking at is a 600-year-old Siberian pine growing at the top of the mountain, just below the alpine tundra. And so we went to that site nearly 10,000 feet above sea level, because the growing season is so short and it's so cold, relatively speaking, for August, that the growing season, the growth of the trees is limited by temperature. And so we went there in August. It actually snowed when we were there on that mountain. That was a lovely day, but there were other days when it was snowing. And so we sampled these trees to reconstruct temperature because of its location in the landscape. Growth is limited primarily by temperature. And so that was one of the questions someone gave me. How do you know when a narrow ring is due to cold or drought? It depends on where you are in the landscape. And at the flip side of this scene, at the bottom of the mountain where the forest meets the steppe, it's hot and dry, and we go there to collect trees to reconstruct drought history. And that's one of the ways we separate what narrow rings might represent in trees. But it's more complicated than that. But this is the simplest approach that we have. Next slide, please. And so I just wanted to show you really quickly. This is really fun. This is research that we're going to do. It's going to be submitted in the next month, I think. And what you're looking at on the x-axis is 800 years from the year 1200 essentially to today. And on the y-axis, we have mean June-July temperature. These data are from Western Mongolia, and they're actually, some of them are from my very first trip to Mongolia in 1998. This is how slow science takes sometimes. The red on the graph is the meteorological data, mean June-July temperatures from thermometers, essentially. And the black line going up and down back through time is the tree ring growth. And so what we do is we correlate the temperatures of mean June, July to the growth of trees in the overlapping period. And then we reconstruct temperature back in the past based on how the trees grew. And so what you can see is, starting around in the mid-1800s, you can see that long upward ramp to where we are today. And this new record, actually, you can't see it on this figure as much, the annual rings, but 1998 was one of the warmest temperatures. In 1998, 1999 was one of the warmest years in central Mongolia. So those first 2 years I was there were actually one of the warmest ones in the last 800 years. But I couldn't have known that because that was my first trip. The trees knew that, and the trees are the ones that showed us now that we were there during exceptional times. Next slide. And so I was really excited. I was a technician at that time, and I was really excited to see climate change firsthand. I mean, you could take the cores out of the trees from the top of the mountains in Mongolia, and you could see that accelerated growth just with the visible eye. You could see that the 600-year-old tree had its widest rings on the outside of the tree, which is the opposite of what we talked about in the chat. You could see that. I was learning climate change impacts directly, and I was excited to pursue it farther because I'm an ecologist, generally speaking, and I wanted to know how climate change was going to change the forest. And so I took that knowledge from what I learned in Mongolia, and I brought it back to the Eastern United States, and I worked in an area that I like where the South meets the North. So that's a major ecotone that runs across the Eastern United States. It's centered around the Hudson River Valley of New York State. The Harvard Forest is laying almost right in the middle of the New England ecotone, where southern tree species to our south thrive, and to the north we have more northerly species. And then over in the Kellogg station area is another place where you have southern trees meeting northern trees. And I wanted to study this ecotone, this meeting of northern and southern forests to try to understand how global warming was changing the growth rates of trees. Long story short is temperature didn't matter too much to these trees because they're all moisture. Almost all of them are moisture stressed. It's getting late in the day. And so my main hypothesis for my dissertation was false. I was looking for temperature and it really wasn't there. And it's mostly about water. Temperature is important to those trees too, but in a less direct way. So next slide. But as a part of my dissertation research, I mentored Mivan Hopton, who you see in this picture, for her honors thesis. And so along the way, I learned that Atlantic white cedar, which is the species that makes up our data nugget, was actually unusually temperature sensitive. Compared to the other 15, 16, 17 species that I looked at that were all drought stressed during the growing season, Atlantic white cedar was the opposite. And so that's the one species that I could study that I was in where I could see the direct impacts of global warming. And so Miv actually did a wonderful undergraduate thesis. This is in Saco Heath up in Maine. It was a lovely autumn day, and she put together a really nice paper that we published that showed that these trees were temperature sensitive, meaning that when it gets warmer, they grow faster. When it gets colder, they grow slower. And our data nugget actually looks very much like her paper that we published in 1995, actually. So I have to give her credit. Next slide. Yeah, thanks. This is Jessie Pearl, who is featured in our data nugget. She started her dissertation just a few years ago. She's completed it. She's now Dr. Jessie Pearl. And this is in Saco Heath again, just a few years ago. And we updated the collection in this site and a few other sites in New England. And that's what makes up your data nugget. We took the concept that's in Miv's paper, and we updated it with the new data that Jessie collected. And we want to know, how are the trees responding to climate? And I think I gave the answer to your data nugget. I didn't think about that too much. I should have thought ahead a little bit more. But anyways, that's the data that you're actually working with. And you're going to see how climate at the large scale is impacting the growth of Atlantic white cedar near a northern range margin. It's a southern species in Maine that reaches its northern range margin. And you're gonna see how climate change in the eastern United States is impacting the growth of these trees. And that's it, I think.
Awesome. Great framing from both of you, Meredith and Neil. So we have a sense of what's going to be going on in both of the great breakout groups, which we're going to be breaking out very shortly. But we wanted Liz to give you a quick update on the graphs that you created with your Conversation Hearts data.
I got it to work. Okay. So here's just some of your graphs. It's a really cool data set now. So we'll definitely keep this up. You can all keep entering data if you want to, and you'll have it from everyone, which is really nice. So yeah, here's just a couple graphs I made really quickly, but you all can play around with it more. So we can see purple is a really rare color, which is interesting. All the other colors seem to be about equal. I can go to another one. This is the phrase data. I had no idea that there were so many phrases on the hearts. I thought there were going to be a standard set, but you can't even read them anymore. So basically there's only one of many different phrases, which is very interesting. And it's not because you all were typing weird either. They're actually different. I was looking at the data set. And then this is our data about the number of letters. So just trying to see basically if like the different color hearts had different phrases or like lengths of phrases on there. So it looks like it's pretty standard. There are a couple like really long phrases. I don't know how they fit 15 letters on a heart, but they all seem to be about like 5-letter words or phrases. So anyway, play around with the data. It's a cool data set now. Oh, Pamela, you're still muted.
Thanks everybody for participating in that. And we're going to now give a try at completing a data nugget in breakout rooms. And so when we go into the breakout rooms, we're going to ask you to turn off your cameras if you want to, and definitely we are asking you to be silent to complete your data nugget. Is someone putting the data nuggets in the chat? And Kara is going to break you out. She will assign you your rooms. And then we'll spend the first 20 minutes working on the data nugget individually, quietly. If you have questions and you're stuck, you can write them in the chat in your breakout room. And then at 4:20, we'll be getting all of you back on unmuted to ask questions and to report back and have a discussion with the scientists and educators in your breakout room till 4:50, at which time we will return back to the main room and we'll all have a few words before we break up for the day.
Awesome.
Anything else that Kara or Liz wanted to say before we broke up?
You have printed copies of the data nuggets mailed to you in your care packages, but then I'll also put the links so you have the digital version of the activity in the chat. And then there's going to be 2 rooms for the Springing Forward data nugget because we have 2 levels of it. So there's one written for like elementary and middle school level and one written for high school level. So depending, if you put your preference down in that poll we sent you, you'll get into the room that fits your grade level.
Yes, that is the plan, and I think it's coming together. If you didn't vote, I have made executive decisions and kind of sorted you evenly. I'll be in the main room this whole time, so you'll have the option to come back if something's crazy in your room or you need to ask me a question. Remember that the whole chat travels with your breakout room, but then it becomes separate. The chat is just in your room for that time, and I think that's it. So fingers crossed. Look for a prompt to join your room now.
Looks like it worked. I got mine.
Oh, good. Have fun.